Hi, and welcome back to Chapter 18. This is the first video, Part 1 on adaptive specific host defenses. In this section, we're going to look at how the adaptive immune system provides targeted, long-lasting protection. We'll contrast it with innate immunity, which is fast but not specific. Adaptive immunity is slower to start, but it's incredibly precise, and it's going to improve with each exposure. This chapter is broken into 5 sections. Today we're covering section 18.1, the big picture overview of adaptive immunity. Later sections will go into T cells, B cells, MHC molecules, and vaccines. By the end of this lecture, you should be able to define 4 key terms: memory, specificity, primary response, and secondary response. You should also be able to distinguish between humoral and cellular immunity, understand the difference between antigens, epitopes, haptens, and describe antibody structure and the 5 immunoglobulin classes. Adaptive immunity is defined by 2 hallmark traits: specificity and memory. Specificity means that immune cells recognize unique antigens on specific pathogens, whereas memory means that they respond faster and more effectively when encountering the same pathogen again in the future. That first exposure to the pathogen triggers a primary response. It's slower and weaker, but later exposures will generate a secondary response that is much faster and much stronger, and this explains why vaccines work. On this diagram, the first peak shows the primary immune response. It's low and pretty slow, and we're going to see later that it's mostly response through IgM antibodies. After a second exposure to that same pathogen, notice how steep and high the antibody curve becomes. So that's the secondary response powered by our memory cells, and we're going to learn later that it's dominated by a type of antibody called IgG. Let's summarize the differences here. In that primary response, the IgM antibody is the first antibody produced. That is followed by IgG. It has a slower onset because the immune system is learning about the pathogen. In the secondary response, memory B cells and T cells are going to activate quickly, and we're going to see that IgG antibody levels spike rapidly and reach much higher concentrations. And this is why booster vaccines are so effective. They stimulate this secondary response. Adaptive immunity has 2 coordinated branches. First, we have humoral immunity that's mediated by B cells, and these will produce antibodies that target extracellular pathogens like bacteria and toxins. For cell-mediated immunity, that one is mediated by T cells and targets infected host cells. So our cells that are no longer normal because they're infected by some kind of intracellular pathogen that's hiding inside the cell, such as viruses. T cells can also act as the regulators, so they're going to coordinate and direct the whole adaptive response. This first checkpoint asks, list 2 defining characteristics of adaptive immunity. So 1 is specificity. We target unique antigens on specific pathogens. And the second is memory. The adaptive immune system responds faster and stronger upon re-exposure. For the second checkpoint, they ask us to explain the difference between a primary and secondary immune response. So the primary response is that first response or first exposure to the antigen. It's a slow onset and we have lower antibody levels. IgM antibodies are produced first, followed by IgG. In that secondary immune response, it's re-exposure to the same antigen, so it's rapid and strong. You're going to see a high IgG antibody response, and it's driven by memory cells. For the third question, how do humoral and cellular immunity differ? So humoral immunity is mediated by B cells. B cells are going to produce antibodies. Well, they produce an intermediate cell that's going to produce antibodies that target extracellular pathogens. For cellular immunity, sometimes called cell-mediated immunity. These are mediated by T cells, and they're going to target infected cells and intracellular pathogens, so pathogens hiding inside the cell. T cells will also coordinate and regulate the overall immune response. Antigens are molecules that activate the adaptive immune system. They can be found on bacterial capsules, they can be found on their cell walls of bacteria, fimbriae, flagella, viral capsids, or envelopes of viruses or bacterial cells. Antigens can be proteins, carbohydrates, lipids, or nucleic acids, but proteins are the strongest antigens because of their structural complexity. And this complexity is key because immune receptors bind to specific three-dimensional shapes. So an antigen is the full molecule, but immune cells can only recognize small regions of it. And these small regions are called epitopes, like we see here in this picture. So a single antigen, because it's so large, can have many epitopes, and different antibodies can bind to different epitopes. More epitopes means a stronger antigenic potential, and that's why, or one reason, why proteins can generate such a robust immune response. Again, they're large and complex and usually have a variety of different epitopes. This figure shows us how 3 different antibodies binds to 3 distinct epitopes on the same antigen. So notice how all the antigens look the same, and each antigen has 3 different epitopes on its surface, and the epitope is bound by the antibody, and they're very specific. So here's one antibody with that kind of triangular shape. Here's our second antibody with that kind of square or rectangular shape. And then we have our third antibody with a circular shape. So this highlights the precision of adaptive immunity. We also have molecules called haptens, and these are very, very small molecules that are not antigenic when they're alone. They can only trigger an immune response when they're bound to a larger carrier protein. So examples of haptens include urushiol from poison ivy, which can cause contact dermatitis, or penicillin, which can cause severe allergic reactions in some individuals. And these responses occur because the hapten protein combination becomes a recognizable antigen. This next checkpoint question asks, what is the difference between an antigen and an epitope? So an antigen is the whole molecule that triggers an adaptive immune response. But the epitope are small specific regions on the antigen that antibodies, or we're going to see later on T cells, can actually recognize and bind. The second question asks, what factors affect an antigen's antigenic potential? So one is molecular complexity. We said that proteins, which are the pretty much most highly complex of those antigens, are the strongest antigens. Carbohydrates are okay, they're moderate, and lipids and nucleic acids are pretty weak unless they're combined with proteins, because lipids and nucleic acids are very similar between us and the potential pathogens like bacteria and viruses. For another factor that can affect their antigenic potential, we could think about size. So larger molecules have more epitopes. That would lead to a stronger response, as well as their structural shape, their three-dimensional shape or configuration. The last question asks, why are haptens typically not antigenic, and how do they become antigenic? So we said earlier that haptens are too small. They're too small to trigger an immune response on their own. They only become antigenic when they bind to a larger carrier protein, forming a conjugate antigen. So these are important. These distinctions between all of these checkpoint questions are important to show why some substances trigger a stronger immune response while others do not. We've seen antigens so far. Now let's look at the antibodies that bind to antigens. Antibodies are also called immunoglobulins, or the abbreviated version, capital I, lowercase g. They have this classical Y shape made of 2 identical heavy chains. Here it's shown in purple. and two identical light chains, shown in green, which are connected by disulfide bonds. There are two key functional regions to antibodies. The first is the top half over here, the FAB region, which stands for antigen binding fragment, and this is the part that will bind to antigens. We also have the FC region, that stands for crystallizable fragment, that will determine the class of the antibody and also be the region that interacts with immune cells. So understanding these regions helps to explain how antibodies neutralize and eliminate pathogens. This image shows both the simplified 4-chain diagram and the 3-dimensional diagram of an antibody, a specific class or type known as IgG. If I look more closely at the FAB region and the FC region, I'm going to put a line, oh, it's not a perfect line, but it's like over here. You can see that the top half of the antigen binding region is called the variable region, just from here to here. That is going to change. It's going to be different from one antibody to the next because it determines the epitope specificity. They are very specific to what they're going to bind to. Whereas if I change my color, I'm going to look down here. So spanning the entire constant region, but also about half of the antigen binding region, the constant region dictates the class of the antibody. So that FAB region is the antigen binding fragment, and it contains the variable region that physically binds to the epitope. And the function of the FAB region includes neutralization, that is going to block or prevent the pathogen from attaching to our cells. Agglutination will help clump microbes together. And something called ADCC, which stands for antibody-dependent cell-mediated cytotoxicity. This is going to flag pathogens that are too large for phagocytosis. So this region is all about pathogen recognition. The FC region, or the crystallizable fragment, is going to determine the antibody class, also sometimes called the antibody isotype. This is going to bind to phagocytic cells through FC receptors, and that will enable opsonization. Remember, opsonization is when you coat the pathogen, making it easier for phagocytosis to occur. This can also activate the complement protein pathway, and we'll see which antibody class is best for that. So while the FAB region determines or finds the pathogen, the constant region, including all of the FC region, as well as a portion of the lower half of the FAB region, will recruit the rest of the immune system to destroy the pathogen. This checkpoint asks, describe the different functions of the FAB region and the FC region. So the FAB region, more specifically that variable region, binds to specific epitopes, and it's responsible for, we're going to learn about these functions soon, but neutralization, agglutination, and ADCC targeting. Whereas that lower region, the FC region, that constant region, determines the antibody class, and we're going to see soon that it binds to phagocytes, natural killer cells, and complement proteins. And this is going to mediate opsonization and complement activation. So when we think about how FAB and FC differ, one region recognizes the pathogen, whereas the other recruits the killing mechanisms. So that FC region determines the class or isotype of the antibody, and now we see the 5 different antibody classes. So IgG is the most abundant class, and these are able to cross the placenta, they can protect the fetus during childbirth, and they're very versatile. IgM are the antibodies that are first produced, and we're going to see soon that they have a pentamer, or they can produce a pentamer structure that makes it great to agglutinate pathogens, clump them all together. IgAs are found in our body secretions like mucus, saliva, tears, and breast milk, and it protects our mucosal surfaces. For IgD, they are going to be attached to the surface of B cells as a membrane-bound receptor. They are not secreted. They're also probably the least well understood class. Then we have IgE. These are involved in parasitic defense. They can also be involved in allergic responses through mast cells and basophils. All right, so we can see the structure of each of the antibodies here. Pretty typical of what we saw earlier, except for IgM can form that pentamer I described earlier. So that makes it really good at agglutination, bringing these pathogens together to clump them up. IgA, you can see, produces a dimer, so two of these antibodies linked together. If I look at the heavy chains, that constant region, that determines the antibody class. Excuse me, these are Greek letters. So we have gamma for IgG, we have mu for IgM, alpha for IgA, delta for IgD, and epsilon for IgE. And if I look at the most common, so looks like the most common antibody that's present in the blood serum is IgG. Also notice which antibodies fix complement proteins, which can cross the placenta, and how many antigen binding sites they have. Most of them have 2 antigen binding sites, but the IgM, you can see, can have 10. The first checkpoint question asks, what part of an antibody molecule determines its class? So we saw that it's the constant region of the heavy chain of the antibody. And we had the gamma, that is IgG, mu for IgM, alpha for IgA, delta for IgD, and epsilon for IgE. For the second question, what class of antibody is involved in protection against parasites? We saw that this was IgE, right? IgE can bind to mast cells and basophils, and that will release inflammatory mediators. Finally, describe the difference in structure between IgM and IgG. So IgM, we saw earlier, is secreted as a pentamer. There were 5 antibodies essentially joined together. And because there were 5 of them and each one has 2 binding sites, they have a total of 10 antigen binding sites. Whereas in IgG, we have a monomer here, that typical Y-shaped antibody with 2 antigen binding sites. And this one is the most abundant in the blood serum. All right, so now let's look at what happens when antigens and antibodies interact. What is the result? One potential reaction is neutralization, and neutralization occurs when antibodies, especially IgG, IgA, or IgM, binds to antigenic sites on pathogens or toxins and physically blocks them from binding to host cells, essentially our cells. So this is particularly important for viruses because blocking attachment stops infection before it even begins. Here we can see antibodies binding to a toxin, preventing entry or preventing it from binding to our cells. And here's another example of, looks like it's binding to a flagellum. The second type of antigen-antibody interaction that can result is opsonization. Opsonization, actually it means something funny. It means to make tasty. So in our case for the immune system, it makes it easier for phagocytic cells to eat the pathogen, right? To ingest it and digest it. So what you're going to see is that IgG can bind to a pathogen, and the constant region, remember that bottom part of the Y structure or Y shape, is going to bind to Fc receptors that are found on phagocytes like macrophages, dendritic cells, or neutrophils. And this dramatically increases phagocytosis efficiency. Our third type of interaction results in agglutination. So agglutination is the clumping together of pathogens. And IgM is the best at this because of its pentamer structure. Pentamer refers to 5, so you can see 5 of them linked together. That offers 10 binding sites on the surface of these pentamer structures. So these clumps of pathogens make it easier for like our spleen and our kidneys to filter and easier for phagocytes to consume. Antigen-antibody interactions can also lead to complement activation. The classical complement pathway is triggered when either IgG or IgM binds to a pathogen or pathogens. This leads to inflammation, enhanced opsonization, and formation of that membrane attack complex, which lyses gram-negative bacteria. Remember, this generates large holes in the bacterial cell, which causes water to rush in and lyse these cells. Finally, our fifth and last reaction is our antibody-dependent cell-mediated cytotoxicity. So ADCC targets pathogens that are too large to be phagocytosed. So what we're going to see is the FAB region binds to the pathogen. So you can see that in all of these examples. And the immune cells, like natural killer cells or macrophages or eosinophils, are going to bind to the constant region, the FC region of the antibody. That activates these cells, and these cells are going to now release molecules like perforin or granzymes that kill the target cell. All right, this checkpoint asks, where is IgA normally found? So IgA is normally found in mucus and mucosal secretions, like the respiratory, GI, or urogenital mucus. This includes saliva, tears, or breast milk, and it's usually secreted as an IgA dimer, where we saw 2 of those antibodies it's hard to show, like linked like this. The second one, which class of antibody crosses the placenta, providing protection to the fetus? So the only one is IgG. IgG can provide passive immunity to the fetus. The third question asks, compare the mechanisms of opsonization and antibody-dependent cell-mediated cytotoxicity. So for opsonization, it's when IgG binds to the pathogen. The constant region, that FC region, binds to phagocytic FC receptors, and this is going to enhance engulfment by phagocytes. For the second one, ADCC, that antibody-dependent cell-mediated cytotoxicity, this is when the target is too big to be ingested to phagocytose. So the IgG is going to bind to that giant target. The constant region, the FC region, is going to bind to either natural killer cells, macrophages, or eosinophils. And these cells will release cytotoxins like perforins or granzymes to kill the target. All right, let's recap 18.1. Our first learning objective asks, define memory, primary response, secondary response, and specificity. So memory is the adaptive immune system's ability to remember a previous pathogen and mount a faster, stronger response upon re-exposure. And that primary response was triggered by the first time exposure to an antigen. It was slow and resulted in lower antibody production, and we saw that IgM appears first. But the secondary response was based on subsequent exposure to that same antigen, and it was rapid, high-level antibody production that was dominated by IgG antibodies and due to memory cells. Specificity refers to the ability of the adaptive immune system to target unique antigens that are associated with specific pathogens. All right, the second learning objective asks us to distinguish between humoral and cellular immunity. Humoral immunity is mediated by B cells. We're going to see that they're going to produce antibodies that target extracellular pathogens or toxins, and their functions will include neutralization, agglutination, or opsonization. Whereas for cellular immunity, it's mediated by T cells. They're going to infect or target infected host cells and intracellular pathogens, and they're going to coordinate immune responses and also kill infected cells directly. The third learning objective asks differentiate between antigens, epitopes, and haptens. So antigens are entire pathogen-associated molecules that trigger an adaptive immune response, like proteins, carbohydrates, lipids, or nucleic acids. Epitopes are small specific regions on an antigen that antibodies or T cells actually bind. And one antigen, because it's so big, can have multiple epitopes. For haptens, they were too small, right? They were very small molecules that were not antigenic alone. They only become antigenic when they're attached to a larger carrier protein. And we saw examples included penicillin or urushiol allergies. For the fourth learning objective, they ask, describe the structure and function of antibodies and distinguish between classes. So we saw the basic antibody structure was a Y-shaped molecule made of 2 heavy chains and 2 light chains, and they were all held together by disulfide bonds. There was an FAB region that was the variable region, and they bind to specific epitopes and result in neutralization, agglutination, or ADCC targeting. For the FC region, that was a constant region, and that determined the antibody class and can bind to complement proteins or FC receptors on immune cells. For the different classes of antibodies, we saw IgG were monomers and most abundant in the blood plasma, and these could cross the placenta. IgM can form those pentamers. They're the first antibody that's produced, and they're great at agglutination. IgA, they form that dimer, and they're found in our secretions like saliva, tears, or breast milk. They protect our mucosal surfaces. IgD are attached to B cells, and IgE are involved in parasitic defense and allergic reactions because they can bind to mast cells and basophils. That takes us to the end of section 18.1. In the next video, we're going to continue with antigen presentation and the major histocompatibility complexes.